Hello students, in today's class we are going to learn about microbial nutrition. So microbial nutrition is basically about supplying the cells with the chemical tools that are required to make monomers. Different microorganisms have different set of requirements of nutrients. These nutrients can be broadly categorized into macronutrients and micronutrients. Macronutrients are those that are required in more amount, while micronutrients are those that are required in less amount but are important for cellular function. Examples of macronutrients include sources of carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus, which are components of lipid, proteins, carbohydrates, and nucleic acid. While micronutrients majorly includes metals, which play a structural role in various enzymes. For example, iron, which is component of cytochromes, enzymes such as catalase and iron sulfur proteins. While molybdenum is a cofactor for enzymes like nitrite reductases. Some microorganisms have special requirements that reflect on their morphology or environment. For example, diatoms need silicon or silicic acid. Growth factors are another important requirement, and similar to micronutrients, they are required in less quantity and only by some cells. It includes vitamins, amino acids, purines, and pyrimidines. Most microbes are able to synthesize most of these growth factors. However, some microbes may require one or the other nutrient in the preformed form. Vitamins are mostly part of coenzymes. For example, riboflavin, which is a part of or precursor of FMN, and biotin, which is required in fatty acid biosynthesis. Next, we'll discuss about sources of the macronutrients. So, firstly, carbon. So, most prokaryotes require organic carbon, which is used to make amino acids, fatty acids, organic acids, or sugar. Some microbes can also make use of complex forms of carbon, such as pesticides and oils. Next is nitrogen. It is the next most abundant element in the cell after carbon, and it can be acquired from organic as well as inorganic sources. It is required for synthesizing amino acids, purines, and pyrimidines. Some microbes can reduce nitrite to ammonia, while others can also assimilate atmospheric nitrogen, such as cyanobacteria. Next is phosphorus, which is required for nucleic acids, nucleotides, and cofactors. It can also be obtained from inorganic as well as organic sources. Next is sulfur. It is required because of their structural role in amino acids, cysteine, and methionine, and also in vitamins such as biotin and thiamine. Sulfur also undergoes a number of transformations in nature, mostly because of the oxidation and reduction reactions performed by microbes that leads to formation of SO4 and hydrogen sulfide, respectively. Potassium is also required by all organisms as it is component of some enzymes. Magnesium is required for stabilization of ribosomes. Sodium is also required by some but not by all microorganisms. Mostly seawater or marine microorganisms have a requirement of high sodium content, while freshwater bacteria do not need sodium for their growth. Next, iron plays an important role in several cytochromes and iron sulfur proteins, which are important components of respiratory electron transport chain. Many bacteria will synthesize and secrete siderophores, which are basically iron binding proteins that function to solubilize and transport iron to the cells. Talking about culture media, so these are nutrients that are used to grow microorganisms in laboratory, and they can be categorized into two types: chemically defined media and complex media. So chemically defined media are those media which contains purified components. So we exactly know what is the composition of each component or what is the amount of each component in the medium. While complex media are chemically undefined media, where the exact composition or exact amount of each component is not known. For example, in nutrient agar, we understand that it contains peptone and beef extract. However, what is the exact composition of peptone or beef extract is not known. Then types of cultural media can also be classified into selective and differential media. So selective media are media that will selectively allow growth of certain microorganisms while inhibit the growth of others because of presence of certain components in the medium. For example, if a medium contains lactose as a sole carbon source, then it will allow growth of only lactose fermenters while inhibit the growth of lactose non-fermenters. Then a differential medium is a medium which will contain some sort of an indicator that is added to the medium, which will allow to differentiate between the organisms based on the various chemical reactions they perform during growth. For example, EMB medium or eosin methylene blue medium. So this is an example of both selective as well as differential medium. So it contains methylene blue dye, which inhibits the growth of most gram-positive bacteria, and hence it is selective for growth of only gram-negative bacteria. Also, it contains glucose and lactose as energy source. So those bacteria that can ferment lactose, they are known as lactose fermenters, which will generate acid because of the fermentation reaction. And their colonies appear as dark with black metallic sheen because of the precipitation of the eosin component of the medium. Examples include E. coli. While those bacteria which are lactose non-fermenters will instead make use of glucose, and the end products are not acidic. As a result, they will make translucent or pink color colonies, for example, salmonella. Next, we'll discuss about nutritional classification of microorganisms. So based on the energy source that microbes use, they can be classified into two categories, phototropes and chemotropes. So phototropes are those microbes that make use of light as a source of energy, while chemotropes make use of chemicals as a source of energy. Further, based on the carbon source that they make use of, they are categorized into photoheterotrophs that make use of organic compounds as a carbon source, and photoautotrophs that make use of carbon dioxide as a carbon source, means they can fix their own carbon. Similarly, chemotropes can also be categorized into chemoheterotrophs and chemoautotrophs. Examples of photoheterotrophic organisms include green non-sulfur bacteria, purple non-sulfur bacteria. Photoautotrophs, depending on whether they are using water to reduce carbon dioxide, they are divided into those that use water for reducing carbon dioxide, and in the process they will generate oxygen as an end product. Hence, they are known as oxygenic phototrophs. Examples include algae, plants, and blue-green algae. While there are some which do not make use of water, and instead they will make use of other compounds such as hydrogen sulfide, and as a result, these microbes are known as anoxygenic phototrophs, and they carry out anoxygenic photosynthesis, for example, purple and green sulfur bacteria. The chemoheterotrophs, also based on the final electron acceptor that they are using, it can be using oxygen as the final electron acceptor. For example, in case of animals, fungi, protozoa, as well as bacteria, or rather aerobic bacteria, or instead of oxygen, they can be using certain other compounds, such as they can be making use of organic compounds, in which case they have a fermentative type of a metabolism, for example, streptococcus, or they can be using other inorganic compounds in which they show anaerobic respiration properties, for example, Clostridium species. Uh, the chemoautotropes includes hydrogen, sulfur, iron, nitrogen, carbon monoxide oxidizing bacteria. So this is about nutritional classification of microorganisms. I hope you like the contents. Kindly like, share, and subscribe to get direct updates on the uploaded videos. Thank you.